Hello everyone, welcome to the 5th International Eurasian Conference Biological and Chemical Science. Thank you for this opportunity that given by the organization to present my work as a video presentation. My name is Wasan Meftoul. I am an MC student from Baghdad, Iraq in Ond Kuzmayis University, Turkey. Today I will present a part of my work which includes a novel actinobacterium Arthrobacter species H14-L1 synthesized silver nanoparticles, supervising by Dr. Hilal I. Schedule of today's presentation starts with introduction, objective, methods, result and discussion, and finally, conclusions. Introduction. Actinobacteria constitute one of the essential microbial groups in biotechnological applications. Most members of this group can synthesize a diverse array of bioactive metabolites and enzymes with potential applications in medicine, agriculture, and industry. These bacteria also have importance for the biological synthesis of nanoparticles such as silver nanoparticles. Silver nanoparticles are one of the metallic nanoparticles that are mostly utilized due to their low cost and unique properties. The large surface area authorized to be nano-sized can change the physical, biological, and chemical properties for the purpose increasingly applied. Due to their novel properties that are exhibited in nanoparticles, enabled them to interact with plants, animals, and microbes, which makes it a good candidate used in various fields. The objective of this study is the Arthrobacter species H14-L1, a cyclotorulent actinobacterium isolated from the Antarctica in the previous study, was shown to synthesize silver nanoparticles. The physical-chemical characterization of the synthesized nanoparticles was achieved by using UV visible spectroscopy and scanning electron microscope EDX analysis. The whole genome analysis of Arthrobacter species H14-L1 was conducted to reveal possible metabolic contributing to the synthesis of silver nanoparticles. The genome annotation performed on Rust server to identify protein encoding rRNA and tRNA genes. Then the phylogenetic analysis conducted on the TYGS server helps to classification and identification of prokaryotic strains without overestimating phylogenetic confidence to determine secondary metabolized biosynthetic gene clusters encoding by the genome of H14-L1, the anti-smash server was used. Methods. In order to prepare silver nanoparticles and synthesizing them, the strain H14-L1 is stimulated in 30 ml of terepene soy broth by enucleated them and incubated for 3 to 4 days as shown in the first image above. Following that, the seeding culture by taking 5 ml of the stimulated H14-L1 into and transform it into 100 ml of TSP. Let it also in incubate shaker for 3 to 5 days. The culture broth were centrifuged at 8,000 rpm for 20 minutes, 4 degree, and the supernatant was collected for nanoparticle synthesis. To start the synthesizing nanoparticles, 2 millimolar of silver nitrate solution was mixed, of course, with the same volume of culture supernatant and stirred constantly. Supernatant silver nitrate solution were placed in a shaker incubator 150 rpm. Then the synthesis of silver nanoparticles was observed within a week by color change from light brown into a brown black. If you can see in these two images, E1 represent the supernatant silver nitrate solution before incubating within a week. You can see the synthesized silver nanoparticles and the change in color. The physical-chemical characterization of the synthesized nanoparticles was achieved by UV visible spectroscopy and scanning electron microscopy DX analysis. The whole genome analysis of Arthrobacter species H14-L1 was conducted to reveal possible metabolites contributing to the synthesis of silver nanoparticles. The genome annotation was performed on Rust server. The secondary metabolized biosynthetic gene clusters analysis was conducted on the anti-smash server, and the phylogenome analysis conducted on the TYGS server. Results: UV visible spectroscopy help us to analyze silver nanoparticles by measuring the amount of discrete wavelength of UV or visible light that are absorbed by or transmitted through a sample in comparison to a reference or a blank sample. If you can see here in this image that silver nanoparticles gave a peak between 400 and 450. They are lies under the normal range of silver nanoparticles, which indicate that these silver nanoparticles are reliable to consume. Silver nanoparticles were collected by microcentrifuging, washed with sterile distilled water, then the silver nanoparticles dried and ready to be analyzed. They were examined by using scanning electron microscope, which is producing images of a sample by scanning the surface with the focused beam on electrons. If you can see, these two pictures belongs to nanoparticles. The purpose of SIM analyze is to determine the size of nanoparticles as well as the shape of them, and according to these images that nanoparticles gave the spherical shape. The energy dispersive X-ray spectroscopy known as EDX analysis associated with scanning electron microscope help us to identify elements in the sample. Also, this analysis confirmed the presence of silver nanoparticles. You can see here the silver ion weighted 93 as long as with other elements such as sulfur and carbon. The genome analysis, the whole genome of Arthrobacter strain H14-L1 used in bioinformatics analysis. RUS server help us to reveal the annotation of this strain. If you can see here in this image, the overview of this strain, which is represent the general information about it, like the genome name, the domino of it, the size of the genome. Also, if you can focus here on these values N50 and L50. These values represent the shortest and the longest sequence of counting, as well as the number of coding sequence and the number of RNAs. Some genes encoding into RNAs, only into RNAs like sRNA and tRNA. So this strain shown 142 reductase, and if you can see here, there are 4 nitrate reductase that is responsible to reduction of ion silver in nanoparticles. Here in this image, you can see the subsystem information. These are the categories, features that are encoded or related to. You can see here there are 143 cofactors related to cofactors, vitamins, prosthetic groups, pigments, 22 related to cell walls and capsules, 28 to virulent diseases and defense. But mostly the genome dedicated to amino acids and carbohydrates. The TYGS server managed to place H14-L1 in phylogenetic tree to see if it's related or close to other species. You can see here H14-L1 related to Arthrobacter terrara. Also, it is a novel species according to the differential in color that exhibited in here. If you can check GC contact, you can see they are very different in numbers. That is, phylogenetically means they are not related but close as neighbors. For the secondary metabolite biosynthetic gene clusters, anti-SMASH results gave 4 clusters that synthesized with troponin gave a similarity coding with the carotenoid that which is responsible for the pigment. Also, the synthesized with the type 3 polypeptides, beta-lactamone, and transdimensional ribosome peptide. Conclusion: the strain of H14-L1 was effective successfully to synthesize silver nanoparticles under short time and obvious results. The average of nanoparticle size was determined at 58 to 68 nanometer according to SME EDX analysis by measuring using image server. The synthesized silver nanoparticles gave a peak between 400 nanometer and 450, with the maximum at 490 nanometer, resulting from surface plasmon resonance. The strain of H14-L1 encoding 50 RNA coding and 3,834 protein coding sequence, including nitrate reductase genes, which might be responsible for the reduction of silver ions to silver nanoparticles. This strain also has gene encoding resistant to antibiotics and toxic compounds such as fluoroquinolones and mercury. Strain H14-L1 is a novel actinobacterium with the genus Arthrobacter, considered the significance of novel actinobacteria isolated from extreme habit. Strain H14L1 may have unique chemical capability for the biotechnological application. The strain H14L1 has 4 gene cluster encoding for tryptophan, type 3 polyketide, beta-lactam, and ribosomally synthesized post translationally modified peptide. In future study, a transcriptome analysis will be encoding for Klebsiella pneumonia exposed to silver nanoparticles synthesized by Arthrobacter species H14L1 revealed the pharmaceutical potential of these nanoparticles.